Today in this program, we are going to learn about the isolation of the single colonies on the solid agar media. A bacterial culture media is usually made in order to cultivate or grow a bacteria. Since the bacterial cells are very small, it is not possible for us to measure the dimensions of a bacterial cell and tell that the bacterial cell is growing. And in order to see the growth of the bacterial cell, we need a culture media. Culture media, based on their consistency, is of two types. One is a liquid culture media, or it can be a solid culture media. Now, when it is a liquid culture media, we don't add any solidifying agent. But if any solidifying agent is added to any liquid culture media, then it is converted to a solid culture media. Bacterial colony refers to the population of cells which has resulted or which has developed from a single bacterial cell. So, what a colony is? It is basically a clone of cells. That means all the cells in a bacterial colony are of same type or they belong to the same species. So, if we want to see the growth of the microbial cells which are so minute, we basically look upon their colony growth. We call it population study. Not a single cell is looked upon, but we look upon the population of the bacterial cell. Now, isolation of the single colony on the agar media is very important because we want to prepare a pure culture. So, pure culture means a culture which is being made from a single type of bacteria. Now, this importance of the pure culture can be studied very well when we use this culture in the industry. For example, there are many industries which use a particular bacterial strain for the production of antibiotic, or it can be used for the production of say vitamin, it can be used for the production of organic acid or any other pharmaceutical product. For this purpose, we need to store the pure culture. That means a bacteria has to be maintained in a pure culture form. Now, maintenance of the pure culture is again has to be looked upon because very essential to maintain the pure culture of the bacteria. Either it is maintained in a desiccated form, that means we take out the water from the bacterial cells or from the surrounding and we preserve it in the lyophilized form, or it can be preserved in a glycerol stock. That means in spite of water we add glycerol which does not form any crystals, and therefore, the bacterial culture could be stored at -80 or -40 degrees Celsius. Robert Koch in 1881, he took some potatoes, he boiled the potatoes and sliced the potatoes with the help of a flame sterile knife. At this time, microbiology was developing. So, he knew that the knife he has to use has to be sterile, and these boiled potatoes were used as the solid media for the growth of the bacteria. But the drawback was that all the bacteria were not able to utilize the carbohydrate that is present in the potato. And what is this carbohydrate? Present in the potato that is starch. Okay. Starch is a polysaccharide. All the bacteria are not able to utilize the polysaccharide. There are many bacteria which utilizes the monosaccharides only. So, those bacteria would not grow on the potato. So, this was one of the drawback of the Koch's medium. Then we have the Frederick Loeffler. He was an associate of Robert Koch, and Robert Koch at this time was working with Bacillus anthracis, the organism which causes anthrax. Loeffler said that let us make some solid media where this pathogenic bacteria could be cultivated. He came out with a media which was a meat extract peptone media. Now, Robert Koch took the Loeffler's media and he put some gelatin in this media and observed that it was giving a semi-solid state to the media. So, this was the first thing that he added in a liquid media and tried to prepare a solid media. But the problem with the gelatin is the many bacteria are there who are able to digest gelatin. Then those bacteria will not grow in this media. And another thing was, if gelatin is incubated because we are preparing the media, we are going to put the bacteria on this media and we are going to incubate at 37 degrees Celsius, which is a normal body temperature of the human body, and the pathogens infect the human body. So, we have to maintain the temperature of 37 degrees Celsius, and if gelatin is subjected to a temperature of 37 degrees Celsius, it was seen that the gelatin would melt. So, these were the drawbacks with the gelatin. Minoru Taraziemon in 1882, he was preparing a soup in his home and he had some extra seaweed soup. He threw them off, and then the next morning he observed that it was forming a jelly-like substance. This struck his brain and he said that, okay, this soup could be used for the solidification of the liquid media. So, this was a step towards the use of agar, because agar is a compound which comes from the seaweed. So, Walter Hesse, he was a close associate of Robert Koch. Now, Walter Hesse's wife Fanny used to hear the difficulties that they were having with the use of gelatin, with the use of having or with the preparation of the solid media. So, she was the first who said that, why don't you use the seaweed? And it was found that the seaweed contains a very good solidifying agent, and that was agar. Then we had Richard Petri, who was a German bacteriologist. He came out with the discovery of the Petri dishes. For the liquid culture, we used to have the use of the conical flask or the test tubes. Now, after the solid media was found, the scientists were very much worried that how to give the substrate or how to put a shape to this solid media so that the bacteria could be cultivated. So, Petri came out with a dish which was known as Petri dish, which has got two things. One is a lid and the other one is a base, and the lid fits with the base very tightly with a very small opening. The base was where you poured the solid media, and if you allow it to cool down, then it takes a shape of the Petri dish. Okay. Now, we had another revolutionary discovery by Hansen. Now, Hansen, he studied the individual yeast cells. He took the experiment of hanging drop. That means, on the cover slip, he put the drops of the yeast cells which were cultured in a liquid media. At this time, he was not using any solid media. So, he put the liquid media and he chose only those drops which was having only one yeast cell. And he studied that how yeast cells undergo the budding process, how a single yeast cell produces buds and then develop into mature individuals. So, he was the one who was trying to study the growth of the bacteria with the help of yeast cells. But the drawback of this method was the yeast cells are far larger than the bacterial cells. So, the bacterial cells could not be done because we were not able to see a single bacterial cell in the droplet of liquid media. After Hansen came the discovery of Bury's India ink method. Now, in Bury's India ink method, what he did, he stained the liquid culture of the bacteria with India ink. And after he stained it with India ink, he tried to study it on a solid surface. It was these glass slides on which the solid media was poured, and upon this he added the drop of India ink or the culture which was stained with India ink. And then under the microscope he studied the mobility, the binary fission, and how the bacterial cells grow. The drawback of this method was that all the bacterial species are not able to tolerate the India ink. They are either desiccated, they shrink up, or they die. Therefore, they do not remain viable. So, when you want to study the mobility, the growth of the bacteria, the cells have to be viable. Another drawback was that he used a Petri dish, put on the solid media, and the India ink drops were poured on it. Now, the Petri dish have to be kept on the stage of the microscope, and this was a very cumbersome process because continuously you have to study whether the cells are growing, they are moving or not.
Ma'am, I have a question. Between India ink and the Hansen's method, which one is practically best to be used?
India ink method is much better because in the Hansen studied drops which are having the bacterial cells. Now, all the bacteria we know are not visible with the naked eye even if you use a 100x power. Now, in the case of India ink method, if you stain the bacteria, it becomes much visible to the naked eye, and therefore, the staining method is much better in comparison to the Hansen's method. In environment or in natural form, the bacteria are always present in a mixed population, and we have to isolate those bacteria and prepare the pure culture of the bacteria. So, let us study what are the different methods of the preparation of the pure culture or isolation of the single colony. The first method, which is very commonly used for the isolation of the bacteria and fungi, is the spread plate technique. The most important thing needed for spread plate technique is the use of spreader. This is basically made of glass tube or glass rod. Okay, it has got a triangular base and a large handle. This spreader has to be made sterile because it will be used for the spreading of the mixed bacterial culture. We first take a beaker or a cobbler's jar which is full of alcohol. Dip this spreader inside this container which is having 70% alcohol and then burn it over the flame. The next thing is, let the spreader cool down after you have made it sterile with the help of alcohol and the heat. After you open up the Petri plate, have the bacterial culture in a mixed form. In the center of the agar plate, we call it inoculum. When we take the inoculum in the center of the solid agar media, we use a rotating table. If you just rotate this table, you will find that the plate or the Petri plate which is having the agar media starts rotating, and you just take the spreader at one position only. You will find the spreader spreads the liquid culture media all over the surface of the solid media.
Ma'am, I have a question. What are the advantages and disadvantages of the spread plate technique?
The advantage is, when you are spreading a small inoculum over the surface, mainly the aerobic bacteria grows because you are spreading it onto the surface of the solid agar media. Okay. But the disadvantage is, the bacteria in the mixed population, you have got aerobic, anaerobic, facultative anaerobes, facultative aerobes. Now, facultative aerobes or facultative anaerobes, they can grow on the surface. But if we talk about the anaerobes, they will not grow by the spread plate technique. So, this is basically the disadvantage of the spread plate technique. Now, after you have spread the culture media, we just invert the Petri plates and put it inside the incubator at 37 degrees Celsius for overnight, say for 18 to 20 hours. And after 18 to 20 hours, this is the result what we find in the spread plate technique. You can see the isolated colonies of the bacteria. This was a mixed culture. With the help of color here, you can see the two type of bacterial colony we basically find here. The discrete one, the yellow colors, and somewhat hazy or the translucent colonies are the white color colonies. So, from here, if we pick out one of the colony, this is called basically a colony forming unit. This is a colony, that means this has got a population of cells which has developed from a single bacterial cell. So, if you count a colony, basically or indirectly, you are counting the number of bacterial cells. Okay. So, like this, the different discrete isolated colonies can be incubated or they are obtained after the incubation of the agar plate. Streaking means here you don't use a spreader. In this method, we use another apparatus or another instrument which is called inoculating needle or inoculating loop. Now, this inoculating loop, it has got loop at its top. That means a round color structure at the top of this needle, and this inoculating loop, when inserted inside a liquid culture media, forms a thin film of the bacterial culture. And this thin film of bacterial culture has to be spread over the surface of the solid agar media. Now, again, like the spreader, the inoculating loop which you are going to use for streaking has to be made sterile, and this is made sterile by the method we call incineration. That means direct flaming method. You don't add alcohol, you don't wash it, you simply put the needle directly over the flame, make it red hot, and when you find it becomes red hot, let it cool down, and when it cools down, insert it inside a bacterial culture media which is a liquid culture media, and then start for the streak plate technique. Now, streak plate technique is of various types. The first type we are going to learn, this is called a direct streaking method or continuous streaking. In this method, this is basically a diagrammatic picture of a Petri plate which is having a solid agar media. Now, you have the loop which is having the bacterial film, and you start streaking from the top of the Petri plate. In this direction, you start streaking. That means you keep on doing it. You do not take out the needle, just continuously you keep on streaking the plate in one direction only. You do not take out your needle. Directly you are taking the inoculum and continuously you are putting the streak onto the surface of the solid media. The second technique is called the sector streaking method. Sector means distribution of the area into different smaller sectors, that means into different smaller areas. This is a picture of the base of the Petri dish where you have the solid culture media. Now, this base is divided into 4 sectors, 1, 2, 3, 4 or whatever you name it, A, B, C, D. So, after it has been divided into 4 sectors or what we call it quadrants, now you invert the Petri plate. That means you take the surface, this is the base, you take the surface of the Petri plate and you are able to see the sectors. Now, take the inoculating loop which is having the bacterial culture, the liquid media, and start inoculating. What you were doing in the continuous is you were using the whole plate, but in this method use the first quadrant and then taking the inoculum from the first quadrant, go to the second quadrant, then taking the inoculum from the second quadrant, go to the third quadrant, and like this you inoculate all the 4 sectors of a Petri plate. In this way the streaking can be done, and this is called sector streaking method. Once you are done with the first quadrant, you are taking out your loop. You are using the inoculum from the first to the second. Again you take out your loop from second to the third. But in the continuous, you were not taking out your loop. You continuously did the streaking on whole of the plate.
Ma'am, I have a question. Between the continuous streaking and the sector streaking, which method is best to be used?
Both the methods you can say are easy, but the useful is the sector streaking. Why? Because in sector streaking, you are covering a much more surface area in comparison to the one which you have used for the continuous streaking. In this sector streaking, you have divided it into 4 sectors. So, you are covering a much more surface area, and if you cover a much more surface area, that means the inoculum gets diluted more and more. So, the isolation of single colony is much better in contrast to the continuous. So, after sector streaking, we go on to the third type of streaking, and that is called the 4-flame method. This is very much similar to the quadrant streaking or the sector streaking that we have studied, but the only difference in this case is after you are done with the streaking in the first sector, you take out your loop, you burn your loop. That means your loop is again sterile. The bacteria it was having in the first quadrant has been lost after you have made it sterile with the help of the heat. And now with the help of the sterile needle, you go to the second sector. But the inoculum will be taken up from the first sector only. That means the inoculum amount has diluted. In the earlier case, you have not flamed your needle in the sector streaking. That means you have the addition of the inoculum. But in this case, you are getting rid of all the inoculum which was there with the loop, and you are taking only that much inoculum which is there in the first quadrant. So, you make the quadrants like this. You are done with the second quadrant. You again burn your needle and then go on for the third quadrant, and then similarly burn your needle and go on for the fourth quadrant. So, after the plates have been streaked, we now again invert the Petri plates and put them inside the incubator at 37 degrees Celsius, and let us see the colony development after the overnight culture, that means after 18 hours. Here you can see the pictures that the quadrant streaking, see this is the first quadrant, the heavy amount of inoculum present over here. It gets diluted, and in the last quadrant you can see the single colonies have been obtained. So, this is how the streak plate gives a single colony. Now, all these methods, that means the spread plate technique, the different type of streaking that we have learned, they are used for the isolation of the bacteria and fungi. But Actinomycetes have got a different technique for the isolation. In this technique, You have a nutrient agar media, and onto the surface of the nutrient agar media we put a cellulose ester membrane which has got a pore size of 0.2 to 0.45 micron diameter. If we put the inoculum onto the surface of this membrane, the actinomycetes which grow out their filament structure, they will be able to penetrate the nutrient agar media, while rest of the bacteria will not be able to penetrate and they will not grow onto the surface of the agar media. So, after incubation, simply take out the membrane and you will see the growth of actinomycetes only, because they show a mycelial structure. So, whatever we have studied today is basically how to isolate the single colony so that we could develop the pure culture of the bacteria, of the fungi, of the actinomycetes, because they are of economic importance. Many bacteria are there which are used in the biotechnological industry, while on the other hand there are many bacteria which cause different type of diseases and we call them pathogens. So, we have to study the bacteria, we have to identify the bacteria, we have to characterize the bacteria, and for all these we need to have a pure culture, and therefore isolation of the single bacterial colonies is very important for the microbiological studies.